Hello friends, today we are going to discuss the R principles in laboratory animal experiments. Initially there was 3R principles and then after it was suggested to introduce 2 more R's, and what are that R? Let's see. So that 5 R's are nothing but the replacement, it is the first R, reduction second R, refinement third R, rehabilitation fourth R, and responsibility is the fifth R. All these 5 R's are developed in favor of reduction in or minimum use of number of animals during the experimentations. Now these are 3R principle was first given by the Russell and Birch, and they have treatise, means they have documented this 3 R's in the book and the document entitled The Principles of Human Experimental Techniques on Laboratory Animals, which was published in 1959. So let's see one by one. The first R is nothing but the replacement. The term itself indicates that if you have something good, you can replace that thing, the previous things, with the newer thing. So here in this case, whenever we use the animal, and if you have the another technique to get to perform the experiments, then you can replace the animal with that technique. So it is the replacing the experiments on animal with alternative techniques. There are number of alternative techniques are developed, which includes experiments on cell culture instead of whole animals. So whenever possible, instead of using the whole animal, you can work on the cell culture technique. Then studying human volunteer instead of animals, you can go with the human volunteers whenever it is possible. Then using some epidemiological studies, you can do the research. Then nowadays most popular is the computer models. Develop some computer models which can ultimately replaces the use of animal experiments. Some of the examples are as follows: human volunteers, tissues and cell you can use. Then use some mathematical and computer models to replace the animal. Establish animal cell lines study, and invertebrates like Drosophila, nematodes, worms, they can also be used. So the ultimate goal of this replacement is that if you have some techniques which will give the result similar to the animal experimentation, then you can replace the use of animal and use this in silico or some in vitro techniques. Now second R principle is the reduction. Reduction itself indicate the use of number of animals. It is a quantitative terms whenever necessary. So it is a method that minimize the number of animals used for experiment or test, either by enabling researchers to obtain comparable levels of information from fewer animals. If the researcher is getting the sufficient data by using the fewer animals or few number of animals, then try to use that instead of using the more number of animals, or to obtain more information from the same number of animal. So by this way you can do the reduction in the animal. So it can be carried out by improve experimental design and statistical analysis, shares of data and resources between research group and organization, and use of technology like imaging that enabling longitudinal studies in the same animals. So these are few examples by which you can do the reduction in the number of experimental animals. Then the third important principle of R is refinement. So what is refinement? It is the process of refining the protocol for the quality evaluation and assessment of experimental results. It may include improved protocol with more refined methodology, instrumentation, and experimental procedure. Here, it can be carried out using the less invasive techniques, then better medical care and better living condition. So, for this, use of appropriate anesthetics and analgesic regimens is followed. Avoid the stress by training animal to cooperate with procedures. Then appropriate housing and environmental enrichment that allows species specific behavior. So by performing all these things, one can do the refinement in the experimental animals, and thereby they can get a more appropriate result. Then next R is the rehabilitation. It is also called as reuse of the experimental animal. So most of the time it happens once the experiment is over, the animals are sacrificed. So instead of that, if they if they would use for some other purposes, it can be acceptable. So reuse of experimental animals, if they are not treated with any toxic or harmful or radioactive hazards compounds. If the animals are previously not exposed to toxic or harmful radioactive materials, then you can give the sufficient washing to the animal for a certain period of time, and after that you can use that animal for demonstration like experimental purposes. So this is one of the important part of this reuse of animal. Then the responsibility, the concept of fifth R is most important to be followed by any research fraternity, that is the responsibility for experiments and experimentals animal. So in the new era, there is a need of performance-based outcome of any good protocol, and that includes the scientifically justified objective, then reasonable use of animals, then important is integrity and honesty of a researcher for their work. This is a very important part. Then understand the necessity of life of experimental animal for sound justified outcome. So until and unless there is a sound justification of use of animal, one cannot use the animal. It is the responsibility of the researcher. So this will includes the ethical consideration, then alternative methods of animal experiments and reproducibility of the results, which are again a very important part here we can consider. So keeping in mind all ethical consideration and protest against experiments on animals by several societies, it is always better to know about the alternative of animal experiments and adopt it as much as possible to stop the cruelty or unnecessary use of the animals.